Okay, the recording begins. This is September 19th. It is 2025. This is Biology 105. Thank you all for being here once again. It's so good to see you. All right, cool. Let's go ahead and resume our discussion of nucleic acids. And then we're going to go ahead and get into cell structure, cell function. There are 4 things on the study guide that we have not yet spoken about. They are cell size limitations, differences between prokaryotic and eukaryotic cells, structures and organelles common to both prokaryotic and eukaryotic organisms, and phagocytosis, which is essentially cellular eating. All right, we're going to talk about the components of it and where things end up. Good deal? Nucleic acids. They're going to be super important. We're going to talk about them more and more as we get into our genetics topic. This stuff will come back up. So the nucleotides are the building blocks of the nucleic acids. The nucleic acids, DNA and RNA, are built up of these monomers. Okay? The nucleic acids are composed of a sugar, a phosphate group, and a nitrogenous base. The nitrogenous base is the thing that differs between the nucleotides. Now, DNA and RNA differ slightly. I'll talk to you about how they differ, okay? But within a molecule of DNA, the only difference between each nucleotide will be the nitrogenous base. Within a molecule of RNA, the only difference between each nucleotide will be the nitrogenous base. However, between RNA and DNA, there are some differences, which we will discuss briefly, one of the primary ones being the difference in the sugar's structure. Okay, solid. I don't ask you to remember purines versus pyrimidines. All right, that's not one of the things that we're worried about. But what is important is that some of these nitrogenous bases have single ring structures. Some of them have double ring structures. This is the single ring nitrogenous bases will always pair up with the double ring nitrogenous bases. Third time. A always binds to T. C always binds with G. All right, these are not covalent bonds, they are hydrogen bonds. We will discuss them. A to T is a double bond, C to G is a triple bond, making it slightly more stable. All right, but A always binds with T, check it out. A to T, and C always binds to G, C to G. All right? This is going to set the width of a double-stranded DNA molecule. That's a concept that will come up again later, but I'm going to prioritize getting us through the structure of the biological macromolecule for now, and we'll come back to error checking later. The sugars, okay? The sugars differ between DNA and RNA. In RNA, or ribonucleic acid, it is a ribose sugar that looks like this. In DNA, or deoxyribonucleic acid, it is a deoxyribose sugar, which looks like this. The difference being deoxyribose is missing an oxygen. It does not have this hydroxyl group that is present in ribose. This is the difference between DNA and RNA. Also, there's a nitrogenous base difference, which will come up in a moment. All right? So once you polymerize a nucleotide, okay, once you take a bunch of nucleotides and join them into sequences, you're looking at a nucleic acid or a polynucleotide. These are both terms that are acceptable, all right? And they look like this. Repeating sugar phosphate backbone, all right? Sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. Later in the course, it's going to become important that you can identify the 3' and the 5' end of a nucleotide polymer. What I mean there is a hydroxyl group extending out from the end, or is a phosphate group extending out from the end? The numbering comes from carbon numbering, the phosphate group being attached to the 5' carbon, the hydroxyl group here being attached to the 3' carbon. So looking at this nucleotide polymer right here, we see that it has a 3' end and a 5' end. Cool? This will be important because, well, the main reason it'll be important for you is that DNA synthesis can only occur in the 5' to 3' direction, meaning that as you're growing a nucleotide polymer, you can only add new nucleotides onto this end. That's going to be incredibly important. Okay, awesome. These are all joined together via covalent bonds that are generated via dehydration synthesis. Awesome. Okay, A binds with T, C binds with G, N or G binds with C, and you get a double-stranded structure for DNA. All right? You have a single polynucleotide here and a polynucleotide here. They are complementary to one another. All right? Because the bases need to pair up. What does that mean? Well, base pair complementarity dictates A always binds to T, C always binds to G. So if I split this molecule apart, all right, and I just added the complementary nucleotides, like if I got rid of this strand right here entirely, right, I could regenerate that strand just by adding the complementary nucleotides for each of the nucleotides in this template strand. That's going to be important for DNA replication. So that's base pair specificity, or the DNA strands are complementary to one another, meaning if you separate them, one could be used to regenerate the double-stranded structure by adding the complementary nucleotides. Also, another characteristic, DNA molecules are anti-parallel. Now that can be really confusing, especially with this diagram from your textbook, because when you hear anti-parallel, you probably think, oh look, the sequence goes TGCA in this direction, and on the other strand it goes TGCA in the other direction. That is not what anti-parallel means. It does not mean that they have nucleotides bound in exactly the same order, okay? We can illustrate that, and I know it'd be nice if the marker worked a little better, but just bear with me. A, A, C, C, G, T, the complementary strand is A, C, G, G, T, T. Look, it is not the opposite sequence. If you flip that over, it doesn't align, all right? Bless you. But what anti-parallel does mean is that the 5' end of one strand is opposite the 3' end of the other strand, and vice versa. Good? Once again, that's not something that's going to be incredibly important later when we talk about DNA replication. Look at that, it's anti-parallel. There it is right there. Question, please. This is not on the upcoming exam. Yep, but it'll be so important later. This is not on the upcoming exam, but it will be very important later. Okay, differences between DNA and RNA. DNA is double-stranded, RNA is single-stranded. What are the main takeaways if you're like, want to know why that might matter? Makes RNA way less stable. That's one thing about this, okay? It's incredibly important. They have different sugars, okay? DNA has deoxyribose, RNA has ribose as the sugar. The last difference are the nitrogenous bases that are being used. DNA uses A, T, C, and G, right? RNA uses A, U, C, and G. So most of the nitrogenous bases are the same, but thymine in DNA is replaced with uracil in RNA. Okay? Question. So they both contain genetic code? They both have genetic code. The central dogma is that the DNA is read and it makes a temporary, well, everything's temporary, right? It makes an RNA molecule, which then gets shipped out of the nucleus, where it's read by a ribosome to make the protein. So the RNA is sort of the intermediate molecule, and the stability of the single-stranded molecule, as well as the presence of uracil, has importance for that step of reading this and making a protein, but it'll come back when we actually focus on transcription and translation, those two processes. Solid? I love it. Okay, we gotta mosey, we gotta move fast, you know? So hey, here's one strand of DNA, right? The complementary strand would have this sequence. Because look, the 5' end would be over here, the 3' end would be over here, A to T, T to A, T to A, G to C, C to G, A to T. Question?
So like they're listed out like on the opposite end? You don't just like line it, like flip it around?
Yeah, if I was writing an exam question, which I will do in one of the upcoming exams, you flip them around to make it harder. Because it makes you have to recognize that, oh, the complementary strand, the 5' end, must be opposite the 3' end. That's the anti-parallel nature of DNA, right? So this could be written the other way, right? And this is what the complementary RNA strand would be, okay? Because it has the U's in it, you see. So that does match up. Everywhere there'd be a T, there's a U, okay? Does that make sense? Did I answer that okay?
I just want to make sure the like 3' end goes next to the 5' end.
Yes.
Okay.
Yep, always, like this. Yep. Good deal. It's going to be important, I promise. The directionality of these things is very important. Okay, we don't have time for this. These are extra questions that are interesting, but we just simply don't. Those are like if I'm ahead of schedule. Check them out. They have answers on them, you know? Check them out. Ask me questions about them like during office hours if you're curious about them. Let's do the summary of biological macromolecules table. Macromolecules, the 4 you need to know: the carbohydrates, the lipids, the proteins, the nucleic acids. Look at this as monomer/component. That's because the fatty acids aren't really like monomers because they're not joined in a sequence. Monosaccharides join to make a polysaccharide. That's carb. Amino acids join up to make a protein, a polypeptide, right? Nucleotides join up to make a nucleic acid, also called a polynucleotide. All right? True monomer polymer, true monomer polymer, true monomer polymer, component macromolecule. Okay? Question, please. No, you don't need to memorize the types of linkage. This can be entirely disregarded for the purposes of assignments, exams, and stuff.
Okay, so for this exam, we need to just focus on macromolecule, monomer.
Yeah, this portion right here. Really, it's mainly the macromolecule and the monomer or component.
Okay.
Yeah, of course. Good deal. Okay, there we go. That's week 3's material done. Now we're going to get into the week 4 slides. Okay, is everybody ready? Yes? We're all ready? Okay. Okay. We good, everyone? All right, let's go. Cells. How do we study cells? With microscopy. Cells are small. You need a way to look at small things, make them big so you can look at them, okay? You talked about this in your microscopy lab, right? I believe in the pre-lab video, he showed you light microscopy versus electron microscopy, correct? So we're not going to dwell on it too much. It's fascinating stuff. It's really, really interesting how light has a limit for how much magnification you can accomplish, right? So you use electrons that don't have the same interference patterns. It's all very interesting. Look at this. You get so much more zoomed in. Like all these little purple dots are salmonella in a light microscope. These are salmonella cells in electron microscopy. Boom, boom, there you go. Okay? There are all these different ways of zooming in more or making an image look more clear. It's all very interesting. Light microscopy gets more and more advanced. They're always trying to find ways to avoid using electron microscopy and use light microscopy with computer help, all right? Like doing something such as deconvolution, where you capture an image that's all blurry, but then you use computers to help deconvolute the image and sharpen it artificially. Anyways, it's fascinating. Microscopy is its own field, all right? How small can we get with the different types of microscopy? You can see some cells with the unaided eye, especially egg cells because they're really big, okay? You need light microscopy to see subcellular structures, super resolution microscopy, which is this sort of stuff, okay? You can get to higher resolutions and maybe even see bacteria, viruses, ribosomes, which are organelles, With electron microscopy, can only see actual proteins. We can't visibly see the smallest molecules, and you can't visibly see atoms. So in order to study them, you have to actually carry out reactions, look at what things get produced, measure releases of energy, things like that. All right. This is stuff that I will come back to because it's all really important and it's in your labs, but it's not lab material really more. Question? They can't, they don't. Those are all models. So when you say, like, when you show an illustration of here's a nucleus, here's an electron, that's nothing like the actual structure of the atom. You're just visibly representing there is a cluster of particles that we are calling protons and neutrons, and then there is a probability cloud where we expect that the electron would be located somewhere within this weird shape. So you can't see an atom, we just have models. Yeah. I mean, as far as I know, you can't actually see them. You can see, with electron microscopy, you're looking at scattering patterns of electrons, but things might have changed. They might have them now. Sick. Okay, we'll come back to that stuff. We'll talk about this more honest, because it's very useful and important stuff. All right, ready for the domains of life. We've already talked about them briefly. We've got the prokaryotes, we've got the eukaryotes. Prokaryotic cells versus eukaryotic cells. This is one of the things on your study guide. What they have in common, what's different between them, what organelles are common to each. Okay, so here's your prokaryotic stuff. Ready? Prokaryotic cells, they do not have a nucleus. That's the main thing. Eukaryotic cells, they do have a nucleus. All right? Prokaryotic cells, no nucleus. Eukaryotic cells, nucleus. That's the biggest thing about them, right? Prokaryotic cells still have their genetic material concentrated in the cell in a region called the nucleoid. That's great. There are no membrane-bound organelles. That does not mean that there are no organelles. There are no membrane-bound organelles, but there are organelles present in prokaryotic cells. The organelles that are present in prokaryotic cells are, bless you, ribosomes. Ribosomes are organelles because they are large complexes of different macromolecules. They're made up of protein and RNA, all right? But they don't have a membrane surrounding them. So they still can be present in the prokaryotic cells without breaking this rule. The membrane-bound organelles that we usually consider are the mitochondria and the chloroplasts. Those are our big membrane-bound organelles that are going to be present in eukaryotic cells, or can be present in eukaryotic cells. All right, in a prokaryotic cell, the cytoplasm is the liquid inside. In a eukaryotic cell, the cytoplasm is the liquid inside. The only difference is that in a eukaryotic cell, the cytoplasm is further divided up by the presence of membrane-bound organelles and a bunch of membranes inside of the cell. So they call it the endomembrane system. I'll talk to you more about it, but that is absent in prokaryotic cells. They got their one membrane. A lot of them can have a cell wall, but it's not the same as a plant cell wall. That's it. Cool. Boom, boom, boom. Eukaryotes. All right? Eukaryotes, their DNA is in a nucleus kept safe. That nucleus is a double membrane bound nucleus. All right? The membrane's called the nuclear envelope. It has all these membrane bound organelles, such as mitochondria and chloroplasts and others. Okay? So if a cell has mitochondria, it's a eukaryotic cell. But if a cell has ribosomes, it could be prokaryotic or eukaryotic. Now, you have to have ribosomes because ribosomes are what take the RNA and make proteins. Okay? So if you have DNA, it exists to code proteins. So if you have DNA, but you have no ribosomes, there's really no point in having the DNA. So it makes sense that all cells that use DNA as their genetic code must have ribosomes. Fair enough? Okay, I love it. Size limits for cells. That's on your study guide as well. Okay? One of the primary principles that sort of dictates the maximum size that a cell can be is this concept of the surface area to volume ratio. All right? We'll start with it as a sphere. As you increase the size of a sphere, like as you blow a bubble up or inflate a balloon, okay, the volume inside of the sphere increases more rapidly than the surface area surrounding the sphere. Surface area to volume ratios are incredibly important because reactions occur across surfaces. All right? When we're talking about mitochondria and we're talking about chloroplasts, there are these reactions that occur across membranes, and those membranes are going to be super folded in an attempt to increase surface area to volume ratios. So as you increase the size of a sphere, which for us is modeling a cell, the surface area increases at a slower rate than the volume. Why does that matter? Well, if you've got a bunch of volume, you're going to deplete resources and generate waste within the center of that cell, and you won't be able to export the waste out of the cell or acquire new resources quickly enough because you do not have enough surface area to service the volume. Does this make sense to everybody? Okay, so this creates theoretical size limits for cells if they're spheres, but you can shape cells slightly differently to get them bigger. Like there are cells that go from your head to your toes, all right? And how do they accomplish being so large? Well, because they're cylindrical. So when you're shaped like a cylinder, surface area to volume equations are different. Question, please.
So does that mean that the larger cells struggle to meet like the metabolic requirements because they have a harder time passing the?
That's 100% correct. And I'm just going to repeat it because that's great. Larger cells can have trouble meeting their metabolic requirements because they have less surface area to, get bad stuff out and get good stuff in. Yeah, that's awesome. That's exactly it. So a lot of things have to happen across the surface area of a cell. And we're going to talk about that in a whole lecture that's just called cell membranes. It's very exciting. A question, yes?
Could a large cell sort of be like a sponge with, I guess, holes or tunnels in it that sort of decrease the volume and increase the surface area?
Oh, I see what you mean. Like the cell has like pits in it to increase. So if you think about, theoretically, yeah, right? Like there can be bacteria, single-celled organisms that need to get bigger, and so they can change how much surface area they have through structural proteins that change the shape of their surface area, right? Also, you can change which proteins are embedded in the membrane, and maybe you just get really good at pumping things in and out of the cell so you can get larger. Or you adjust your metabolic demands so that you don't make that much waste and you don't need that much energy throughout the volume of the cell. That can allow you to get larger. So there are single-celled organisms that are shockingly large that you can see and like hold on your palm. All right, there'll be a single cell that just like jiggles on your palm. But they do so by getting around. This is the fundamental sort of physical limitation and the rule of thumb. And with any rule of thumb in biology, there are exceptions to it. And there are many ways to circumvent the physics, right? Solid. Good? Cool. Yes?
I think at one point I learned like some insects are like so small, like some of their organs are just single cells, like they've gotten that small and efficient. Like they sort of reach the limits on like how much smaller they can really get.
Yeah, because now now the organ isn't made up of tissues, the organ is made up of a specialized cell type. Yeah, that's super cool. That's super interesting. You see it, yeah, in single-celled organisms too that like form colonies, and then some of the single-celled organisms will carry on a specific role that services the little colony, like reproduction or photosynthesis or something, or energy storage. So you just scale that up, and now you've got a multicellular organism. That's super cool. I don't know as much about the details there, but I 100% believe it, and it's very cool. I keep saying it's cool. It's because I think it's cool. Yeah. Awesome. Surface area to volume ratios. Feel okay about these, folks? Or any questions about these? Great. It's an important foundational, fundamental concept. Check it out. Cells can increase their surface area without changing their volume by increasing the amount of membranes. All right, fold it up, make, like you were talking about before, making the pockets like a sponge or something. This kind of looks like that, right? So if you need more surface area to fulfill your metabolic requirements, you just need more membrane, you can put it inside the cell. Question?
Do we need to know like the math part of the will our exam have like math issues with the surface area?
No, I wouldn't say like, this cell has a radius of this, what's its surface area, what's its volume? No. Or like, how much does the surface area increase as it goes from this to this? That's like that'd be a fun question, but yeah, I'm not gonna do that. You know what, you've convinced me, I'll put that question on there. Yeah. All right, cool. Man, this is so cool. This is really neat. I found this really cool text. It's there for you to read, okay? Here's a little summary about it. Here it is made digestible, okay? Usually we talk about cells being small to maintain a high surface area to volume ratio. Other factors can influence cell size. Larger cells are also less stable because the membrane can fracture, okay? So that could have something to do. Also, so a lot of the large cells, like muscle cells and neurons that are very long, go from like your brain to your toe, something like that, they'll have a lot of supportive structures within them, like cytoskeletons, we call them, cytoskeletal structures. So that adds stability to them, all right? We're gonna keep talking surface area to volume ratios. You know, it's Bio 105. That's what we talk about. Okay, ready, everybody? Eukaryotic cells versus prokaryotic cells. I want to see how we're doing on the bullet-pointed study guide. I believe we're doing very well. Where is it? Bear with me, folks. Boom. Oh, great. We're doing great. All we have to do is structures and organelles common to prokaryotic and eukaryotic organisms, and then phagocytosis, and then we're done. Very cool. All right, ready, everybody? Eukaryotic cells. Look at all the things inside of a eukaryotic cell. You saw the prokaryotic cell. What does it have in it? Ribosomes, genetic material, concentrated in a nucleoid, but not membrane bound. All right? That's our key difference there. Cool? Good. Eukaryotic cells, okay? Eukaryotic cells, more complicated. They have all of these membrane-bound structures. They have this whole endomembrane system, okay, which allows them to compartmentalize metabolic reactions, carry certain reactions out at much higher rates, keep waste products sequestered. It all just allows for higher efficiency. Look at all this extra surface area inside of these cells. So here's some mitochondria in this one. Is there a chloroplast in this one? No. There's a chloroplast. There's one with a chloroplast coming up. Here's a plant cell. It has chloroplast in it. All right, so eukaryotic cell. Okay, you've got mitochondria. You've got this cytoskeleton, which I already mentioned once before on that previous slide. Cytoskeleton provides structure. It's like a little skeleton for the cell. It's a cytoskeleton. We're going to talk a lot about it. Okay, it's going to be super important later. So we've got Golgi apparatus, endoplasmic reticulum, mitochondria. Okay, these are all special, these are all membrane-bound organelles, and therefore they're eukaryotic specials. Okay, nuclear envelope containing all the nuclear material. Okay, that makes up the nucleus. The nuclear envelope can be contiguous with the endoplasmic reticulum. It's all very interesting. Look at these little folds, these little microvilli on the surface of this cell. Those are designed to increase surface area to volume ratio, indicating that this cell's specialized to absorb something from the environment, probably. But this is just a hypothetical cell. It's also got a flagellum. All right, so it's more complex. Cool? Question? No, you are not. But we're going to learn all of them. We're going to learn them all. And then for exam 2, I might have a question about, like, you know, what goes on in the Golgi apparatus. The only thing that I mention that I have on exam 1 is about phagocytosis and lysosomes. And we've got one lysosome right there. So I'll get to that and we'll talk about that in more detail. Yeah, solid. Okay, listen, cell as a factory. I saw this when I was a student, and I always liked it. It always worked for me as an analogy. But I really want to focus on just getting to the last point of the study guide so we can talk about that. And then we will come back, hit this analogy on the dang next Monday or next Wednesday. Sound like a plan? Cool. Awesome. Awesome. All right. You've got these videos that you can watch. They're linked in here. If I have time, I will play these in class, but currently we don't really have time. So I do recommend you watch these. Sometimes they're very useful for students. They like watching some people you like to watch the video. You can go back, watch it over and over again. You know, it talks about all the stuff in an animal cell. Here's a plant cell. All right, cool. Look at this. One of the key things the plant cell has is a cell wall that surrounds the plasma membrane, the cell membrane. What's that cell wall made out of? Yeah. Cellulose, exactly, which is a carbohydrate. It's a structural carbohydrate, right? Very good points. Yes. It's cellulose. That's its primary, component there. Cool? So remember, it's a structural carbohydrate. It's still a carb. It still has a bunch of energy in it, but we're not really good. Animals don't have the enzymes necessary for breaking it down for energy. They have to make symbiotic relationships with microbes in their guts to break it down. Remember all that? Sounding familiar now? Awesome. Here's a couple of other differences. Chloroplasts. Chloroplasts are the organelles that contain chlorophyll, a pigment, right? And special membrane structures. Look, they're all folded up like stacks of pancakes. It's called the thylakoid membrane. We'll talk about it, don't worry. Okay, but they got the chloroplasts for photosynthesis. They also still have mitochondria, all right? It's easy to forget that, okay? That plants have chloroplasts and mitochondria. Animals don't have the chloroplasts, they just have mitochondria. Because what the plant does is sort of this. It makes sugars inside of its cell, and then it burns the sugars for energy. And you might wonder, why doesn't it just use the energy it took to make the sugar instead of going through an intermediate step? But we'll talk about that when we get to our photosynthesis lecture. It's a good question to be thinking about, though. Tour of a plant cell. Okay, last thing is central vacuole. Okay, central vacuole is just a big vacuole. What's a vacuole? It's a membrane-bound structure. All right, it's a big membrane bubble. It's not like an organelle, really. It's just a membrane bubble that has some stuff in it, some fluid in it. It doesn't have maybe as specific of a function, as much complexity, but it's a little bubble made up of a phospholipid bilayer, right? So the great thing about vacuoles and vesicles, which are small vacuoles, is they can sort of fuse together and split off between all the different members of all the different components of the endomembrane system. Now, the central vacuole is a plant-specific structure that's involved in generating internal pressure to give the cell stability and structure. Plant cells try to stay swollen up. They try to stay turgid so that they have structure. It's like if I took a plastic glove, theater of the mind, here we are. I take a plastic glove, I whack it on my hand, right? It's all floppy, okay? Imagine this is a wilted leaf. Then I take the latex glove and I blow it up and it goes like that. And now it's a leaf that has adequate water. You've seen this with plants before. So what's happening is the plant cells are developing internal pressure and they're pushing against the cell walls, and that gives the larger herbaceous structure stability. Turgor pressure is incredibly important. You are doing an osmosis lab next week where you're going to talk about this concept, and you used osmotic pressures this week to break open cells, right, and release E. coli cells and release their protein. The reason that you could explode them is they didn't have a cell wall to keep them all stable and give them structure. Cool. Question?
What would a fungal cell look like the other kingdom of
Yeah, totally. It looks like more like a tube, all right, like a cylindrical cell, longer cylindrical. It has a peptidoglycan cell wall, if I'm not mistaken. No, a chitinous cell wall, all right? And so the cell wall is a different structure, but they have cell walls. And then the cells are more freely joined to one another, kind of similar to this, where you've got these pores that connect adjacent cells. These are called plasmodesmata in the plant cells, but in the fungal cells, they're called dollipore septa. All right, and there'll be usually, I forget if it's one or many between each adjacent fungal cell, but it's like this tightly regulated, it's a septum, you know, it's got with two pores in it, a dollipore septum, that can allow things to move between the adjacent cells.
So they're basically very similar to plant cells, except instead of the plant structural carbohydrate cellulose, you have the animal one, which is kind of...
They're way more close to animal cells, actually. They're closer to animal cells, but they have a cell wall. And yeah, no chloroplast. No chloroplast. Fungal cells do not have chloroplasts. They're all heterotrophic organisms, though they can form symbiotic relationships with other species that have chloroplasts. But the kingdom fungi, right, does not have photosynthetic members. They're all heterotrophic. They all have to eat. Yeah. Awesome. And then, of course, there's going to be variability between multicellular and unicellular fungi, but I don't remember that much about it if I keep going, yeah.
So can plant cells also have a cytoskeleton and microvilli?
Yes, they can't have microvilli, but they can have a cytoskeleton.
Okay, microvilli is just for animal cells?
Yeah, man, you know, there's probably an exception. There's probably a plant cell that does have that to absorb things from its environment better. And it could be, especially if you're talking about like cyanobacteria, you know, single-celled plant-like structures. So there's going to be some exceptions, but I would not worry about that as a key factor in delineating between the two.
Makes sense, thank you.
Yeah, of course. Okay, hey, shh. All right, ready to keep moving forward? Sorry. All right, plasma membrane. We've talked about it already. It's the primary barrier for movement of substances, okay? Membrane-bound organelles, nuclear envelope, outer membrane of a cell. It's all the plasma membrane. It's made up of a phospholipid bilayer that has some stuff embedded in it, all right? Awesome. Cool. We're going to talk a lot about this. We have an entire lecture on membranes, all right? So the plasma membrane, the cell membrane the cell membrane is what we call it when it's got all this extra junk embedded in it. It's all very important, okay? Some of these are proteins that allow things to move. Some of these are proteins for cell-to-cell recognition or anchoring things. I'm just making sure that there's nothing in my study guide about... We're doing well here. Okay, this all looks good. Okay, great. Let's go ahead and get into, we'll come back to it, I promise. It's all very important. It's all very fascinating. I keep saying that. Nucleus contains the nuclear material, the genetic material. Okay, it's a membrane-bound system. Every single one of these is made up of phospholipid bilayers that have stuff embedded in them. Nuclear envelope, outer layer, inner layer. It's got holes in it to export RNA as well as to do other things. Well, there it is, a nuclear envelope. It looks so intimidating, okay? It's contiguous with the endoplasmic reticulum. We'll come back to it. What we really need to do, though, is get to here we go, phagocytosis. So the endomembrane system, it's a bunch of membrane sacs that are all connected by vesicles, okay? And these vesicles can fuse with other members of the endomembrane system because they're all phospholipid bilayer. So two vesicles or two vacuoles, which are large vesicles, can go together and fuse together, just like two droplets of oil in water joining together. Bless you, right? How's that concept for everybody, okay? So the cell's going to move things between components of the endomembrane system by packaging them into vesicles and then shipping that vesicle off and letting it fuse with another member of the endomembrane system. Like so. Now, you'll see right here, lysosomes. This is the last thing we have to know before your exam. Phagocytosis is when a cell it encounters something outside of it, all right? Let's say that it's like this, okay? This is the outside of me, I'm a cell. I encounter something that I want to eat, and so I fold in my membrane, and I capture the food inside of this little sac, pop it off, and then bring this into the cell, okay? Now I have what's called a food vacuole. It's a vacuole that holds food. And then I take all these other sacs called lysosomes that are full of hydrolytic enzymes, and those I fuse them, food vacuole, lysosome, fuse them together, and I break down the food product, all right? Now, the only trick here is that it goes like this: food vacuole, lysosome, lysosome. When they fuse together, they're called a lysosome. All right, that's the only trick to this. So here it is. Infolding of the plasma membrane, capture the food vacuole, fuse the digestive enzymes, all right? Allow digestion to happen. Questions? This is a lysosome, and this is a lysosome. Once they fuse together and it's got the hydrolytic enzymes in it, it's a lysosome. Yeah?
Sorry, just to understand. So the cell eats another cell, basically.
It can be a whole other cell if it's small and if it's larger, if the size disparity is there.
So what exactly is it ingesting, I guess?
Yeah, it can be all sorts of things. It can be a big macromolecule. It can be like a protein. All right. It can be a chunk of a cell. It could be something that was already partially digested in the environment. It could be like a soil particle with things on it. It can be an entire smaller organism that it envelops, like what amoebas do, you know.
Why do cells do this?
For food. It's eating. Yeah. Phage relates to eating. All right, so phagocytosis is to get food. You have to get the food into the cell in order to break it down.
So basically, phagocytosis is when another cell eats something, like engulfs it in an envelope, and then has a lysosome and a food vacuole, and they combine,
Oh.
and then get a lysosome.
Yep, they fuse together, and now you have the lysosome that contains food particles and the enzymes. And then it digests the food. Yes, and then I'll get you, and then I'll get you. Yeah. Of course. Was that good that answer?
I think so.
Okay, solid. Yeah, please.
Is it still the food vacuole? If it's just like waste, it's like, if something needs to get rid of, is it still a food vacuole?
Not really. So we have things like autophagy, where you eat your own things, own stuff inside of you, and then I'm sure there's a specific name for it in cell biology, but I don't know what it is. So food vacuole, you're talking about like it's going to break this down for energy, even though when you're doing autophagy and breaking something that's like malfunctioning down, you're going to probably get energy out of it. It's more about breaking the thing down so it stops causing any problems.
Of something else, like iron or something.
Yeah. Yes.
So plant cells do phagocytosis?
It's more tricky, right? Because they have their cell wall. The cell wall is actually the biggest problem. So with a plant, as we think of it, like a multicellular, like tissue plant, like a growing plant, like a tree or a sunflower or something, it's probably not going to be doing phagocytosis, right? But a single-celled autotrophic organism may still carry out phagocytosis, and they do. They're called mixotrophs. So which we looked at one in our very first or second lab, where we saw an organism that had didn't have chloroplasts. It could carry out it has chloroplasts. It has chloroplasts. It can also carry out phagocytosis to eat things smaller than it. But it's single-celled. So single-celled things usually do phagocytosis more than multicellular things. Because multicellular things acquire stuff, differently.
And even if they can like make their own energy via photosynthesis, they can still.
They might supplement. Yeah, they might supplement even if they can carry out photosynthesis. Yeah, because they still have all the machinery for breaking down. They still have mitochondria. They have all the machinery for breaking down the sugars they make. So if you find extra sugars, maybe you just dial back the amount of photosynthesis you do. You save some energy. You save having to do as much metabolism. Yeah.
Can you do the demonstration with your hand?
Yeah, absolutely. All right. So I am the I'm the interior of the cell. What, like this? I did it like this. This is better. Okay. My arms are the cell membrane. Okay. Outside my hands is something that I want to eat. So I fold this in. It's called invagination. You fold in the membrane and you capture the food particle there. Okay. Now it's in a vacuole, and that can float throughout the cytoplasm. It can just float inside the cell. So now I've got this vacuole that's holding food. I take a lysosome, and that has enzymes in it. I pop them together, and now digestion can occur. The big trick is that it's as long as it has the hydrolytic enzymes in it, it's called a lysosome. So lysosome, food vacuole, lysosome,
Lysosome, food vacuole.
lysosome.
So when the lysosome goes into the food vacuole, it just turns into another lysosome.
Yeah, when they fuse together. Huh, what's that?
With energy in it.
Well, yeah. Now it's a lysosome with food in it. It's like, think about like your stomach. Your stomach's your stomach. Is it your stomach with food in it or your stomach empty?
Okay.
Yeah, that's good. Yes.
Is it pretty similar to the process slime molds go through?
Yes.
Is that
They do endocytosis, right? And endocytosis is the general term for whenever you invaginate the membrane and bring something in. So there's phagocytosis, which is cellular eating, and penocytosis, which is cellular drinking. And both processes fall on under endocytosis. Yeah, awesome. We did it, everybody. Good job. Oh, I got one more question. Yes. That's okay.
So do we need to know what happens after it becomes the lysosome and then it's fully digested? Do we need to know what it does after that?
No. Just think that it then is gonna, now it's got a bunch of sugars in it or high energy molecules have been broken down. It's gonna go put them somewhere. It's gonna go feed them to a mitochondrion. Of course. Thank you, everybody. I hope you had a good class. I know I did. And I will see you again on Monday for the exam. Don't forget there's an exam on Monday. Thank you. Okay.
Scantron, right?
Yeah, Scantron 8.